We will discuss the topic protein synthesis. The objective of this topic is to understand the mechanisms of the protein synthesis process and the role of the different RNAs and different factors and the proteins and the enzymes in protein synthesis process. This is the second part of central dogma, that is molecular biology, in which the RNA form the proteins with the help of the different enzymes and the factors. This is the process in which the genetic code in the mRNA is read to make the proteins, and this whole mechanism has been explained in the figure in the next slides. After the mRNA formations, and it has to leave the nucleus and it moves to the ribosomes, which consist of the ribosomal RNA and the proteins. The ribosome read the sequence of the codons in the mRNA, and molecules of the transfer RNA bring the amino acid to the ribosomes in the correct sequences. Each transfer RNA molecules has and the anticodon sequences you all know for the particular amino acids which it carries. An anticodon is complementary to the codon for an amino acid. For example, the amino acid lysine has the codon AAG, so the anticodon will be the UUC. Okay, so with the help of the ribosomal RNA, bond forms between the amino acids, and it bring about on the particular sites on the ribosomes, and it lengthen the polypeptide chain. The chain of amino acids keeps growing continuously until the stop codon reached in translation process. So this is the process in which the ribosomes in the cytoplasm or the endoplasmic reticulum synthesize the proteins after the process of transcriptions of the DNA to RNA inside the cell nucleus. The whole process is called the gene expressions. In the translation process, the mRNA is decoded in the ribosomes outside the nucleus and produce the specific amino acid chain. The polypeptides later fold and convert into the complete functional proteins. The ribosome facilitates and decoding by inducing the binding of the different complementary transfer RNA anticodon sequences to mRNA codons. The transfer RNA carry the specific amino acids which are chained together into the polypeptide chain. The whole process is divided into the three different steps. One is initiation, second is the elongation, third one is the termination. In initiation steps, ribosomes assembles around the target messenger RNAs, and the first transfer RNA is attached as the start codon. The elongations. In elongation steps, the last transfer RNA validated by the small ribosomal RNA subunit, that is called the accommodations, and transfer the amino acid which it carries into the large ribosomal subunit, and it binds it to the one of the proceedingly admitted transfer RNA. That process known as the transpeptidations. The ribosome then moves to the next mRNA codon and continue this process, which is known as the translocations, and that's by the process it creates the amino acid chain. Now the terminations. When the stop codon reached, the ribosome release the peptide chain and the whole process becomes blocked. Now the tools of the transfer RNA. In this process, the following tools are the used, or the components are the used. That is main tools are the ribosomes and the transfer RNA. Apart from this, mRNA, initiation factor, elongation factor, termination factors, and the amino acids are the used. Next is the ribosomes. Protein synthesizing factory of the cell. This is the ribosomes, which are the two types, 70 and the 80S types. And in ribosomes, there are the three different sites are the present: A sites, B sites, and the E sites. And mRNA binding sites is also available on the ribosomes. mRNA binding sites bind the sequence near the 5' end of the mRNA and place the mRNA in the proper positions for the translation process. The binding sites all are located at or near the interface between the large and the small subunits of the ribosomes. So you know this is the structures of the ribosomes, in which you can see the two different parts, small and the large part, that is 30S and the 50S, and complete unit is the 70S ribosomes. And in another figure, see, you can see the different sites, E, P and A sites, in which the process is going on. Okay, and the first codons, that is formylated methionine or the formylated methionine, the eukaryotes, is come and bind first of all and start the process. So the transfer RNA molecules, next component is this is the molecule which contain the three major loops, that is 3' terminal sequences, that is CCA in positions, and where the appropriate amino acids come and bind. And this model is well known and known as the clover leaf model. Now the next step is the charging of transfer RNA. In amino acids, activations process or charging of transfer RNA, also known as the amino acylations process. Amino acid transferase enzyme is used and it binds the ATP molecules to the amino acids, and one pyrophosphate molecule is released in this process. Amino acid transfer RNA synthetase binds the AMP amino acid to transfer RNA, and it is used in the next step. During the amino acid activations, the amino acids are attached to their corresponding transfer RNA. This is the reactions in which you can see the amino acids bind with the ATP and it forms the amino acid AMP molecules, and then this AAMP molecules attached with the transfer RNA, and then AMP becomes again released, which used in the next steps. Now we start the process of the translations. So in this process, the amino acid first is coupled to the penultimate, means one before the last one nucleotide at the 3' end of transfer RNA by the ester bonds. The formations of the ester bond conserve at considerable part of the energy for the activations steps or activation reactions. Each of the 20 amino acids are recognized, as you all know, by its specific amino acid transfer RNA synthetase enzyme. The syntheses are usually composed of 1 to 4 protein subunits, and enzymes vary considerably in the structures, although they all perform the same functions by binding the ATP molecules. The specificity of the amino acid activation is as critical for the translational accuracy as the correct matching of the codon with the anticodon sequence, which are the present on the transfer RNA. The reason is that the ribosomes only sees the anticodon of the transfer RNA during the translation process. Thus, the ribosomes will not be able to discriminate between the transfer RNA with the same anticodon but linked to the different amino acids. That's why editing mechanisms occurs when there is a misactivation of the amino acids occurs, where an amino acid is attached to the wrong transfer RNA molecule. The amino acid transfer RNA synthetase can hydrolyze the amino acids before it attaches to the wrong transfer RNA molecules, okay, for the corrections. The error frequency of the amino acid activation reaction is approximately 1 in the 10,000 molecules, okay. And the amino acid activations was first of all characterized by the scientist Malone Hoglands. In prokaryotes, the synthesis of the proteins and the first codon is modified, that is the N-formyl methionine, and the chain initiations begins with the formations of the two complexes, that is the IF2 and the N-formyl methionine transfer RNA. And the second is the mRNA 30S subunits IF3. The prokaryotic mRNA contains the consensus sequences which known as the Sine-Delgarno sequences. In this figure, you can see the sequences which are represented as the Sine-Delgarno sequences, and these are the complementary sequences of the nucleotides which present at the 3' end of the ribosomal RNA of the 30S subunits, that is the smaller units. The interactions between these complementary sequences enhance the attachments of the 30S subunits to the AUG initiator codon, which start the process. Both the complexes combine with each other IF1 and the GTP molecules, and the 50S subunits, means the large units of the ribosome, get added to the complex, and IF3 is now released, okay, from the complex form. The additions of 50S subunit utilizes the GTP molecule first of all, and which in turn triggers the release of the IF1 and 2, which means now the IF1, 2, 3 all are released. Additions of the 50S to the complex positions N-formyl methionine transfer RNA in the P sites of the ribosome structures directly with the anticodons of the transfer RNA and aligned with the AUG codons of mRNA. With the AUG initiator codon positions in the P site, the second codon in the mRNA positions in such a way that it corresponds to its incoming amino acid transfer RNA at the A site. In this figure, you can see the whole process, how the amino acids comes and binds to the A sites, and after the binding, then the ribosomes moves on the polypeptide chain, and the next amino acid comes and then bind on the P sites. Now the chain elongation steps. In the elongation steps, this begins with the binding of the second amino acid transfer RNA at the A sites. The transfer RNA is escorted to the A site by the elongation factor, that is EFTU, which also carries the wound GTP's molecule. As the transfer RNA binds, the GTP are hydrolyzed and EFTU is now released. A peptide bond formed between the N-formyl methionine transfer RNA at the P site and the second amino acid transfer RNA at the A site, which is catalyzed by the enzyme peptidase transferase. The transfer of N-formyl methionine to amino acid transfer RNA at the A sites, forming a peptidase transfer RNA at that positions and leaving an uncharged transfer RNA at the P sites. The peptidase transfer RNA translocated to the P sites and uncharged transfer RNA is translocated to the E site. A site is unoccupied, then the new amino acid transfer RNA bind to the A site and the process remain continued in this way. Now the termination steps. When the stop codons like the UAG, UAA, UGA, any one of this codon comes and reach near the A sites, it is recognized and the protein release factors are the come and bound on that sites. Then the two classes of the release factor, that is the 1 and 2. In E. coli, class 1 release factors are the RM1 and the RM2 are seen. These factors bind to the termination codon at the A sites and stimulate the hydrolysis of the bond which form between the transfer RNA and the polypeptide chain at the A sites, and it results completely polypeptides releasing from the ribosome. Then the termination is complete by the release of the mRNA molecules from the ribosomes subunits. This is all about the process of the protein synthesis.